Okay, hello. So this is the final part in this webinar on proteins. And what we're going to be talking about today is the different functions of proteins. So we talked about the structure, obviously, in the very first webinar in this chapter. We talked about the amino acids. And so the structure was last time. And now this time we're talking about its functions. So in general, there are 8 major protein functions. Wow, that's a lot. We have messenger functions, which controls activities between the cells. For instance, insulin released from the pancreas binds to receptors on other cell types. Receptors control activities in the cell. For instance, insulin receptor binds insulin on the cell surface, stimulating glucose uptake into the cell. In this particular case, we're talking about insulin, and it's considered to be a hormone. Hormones can be pretty much any molecule, proteins included, and non-proteins, even like the ethene molecule that helps to ripen bananas, right? If a banana starts to get ripe from green to yellow, ethene is produced, and that signals other nearby bananas to begin to ripen. So ethene is obviously not a protein, but is a hormone in that particular case, and here insulin would be an example of a hormone. So the hormones can be whatever is going to be transported and then therefore involved in signaling some kind of chemical reaction, even biochemical, right? Function, transporter. Description, transport nutrients. For instance, glucose transporter transports glucose from the bloodstream into the cell. Hemoglobin, for instance, transports oxygen through the bloodstream to the tissues. Storage, it stores nutrients. Casein stores protein in milk. We mentioned that in the last webinar. Ferritin stores iron in cells. Contraction, including muscle contractions. Myosin and actin make up the thick and thin filaments in muscle tissue. Protection, antibodies designed to bind to foreign substances in the body, identifying them for elimination. Structure, with shape maintenance, collagen supports, for instance, the structure of ligaments, tendons, skin, nails, and horns. Actin is also found in the supportive structure of the cell. And catalysis, whereby biochemical reactions can occur. For instance, pepsin breaks peptide bonds and proteins in the stomach during digestion. All right, so of course these would make for a good exam question, but let's begin. So let's talk about messengers, receptors, and transporters. Here's our cartoon. And again, we have that phospholipid bilayer. You've already seen this before in some of the other webinars. Maybe you've seen some of this in your physiology courses or elsewhere. But in essence, it's the same thing. We have these phospholipid bilayers. We have the inside of the cell, which is inside this convex and concave shape, I guess, depending on your orientation. Outside of it is more of the exposure to the atmosphere. In this particular case, the environment. The environment would be blood, right? Outside the cell, you have the insulin and glucose, and here you have the cell membrane. Inside, you have the glucose transporter, which is synthesized actually upon need. And then you also have insulin receptor, Y-shaped structure. This is all inside the cell. So let's talk about it a little bit more. I just introduced the hormones and said that could be proteins, so it doesn't have to be proteins though, but insulin is an example of that acts as a messenger between cells. It's released by the pancreas and signals the cell to allow passage of glucose. So if the insulin receptor within the cell membrane receives the insulin molecule, then glucose can be transported provided the integral membrane protein can be produced. So here, this is synthesized in response to insulin, and it does so in order to admit then the glucose from outside to inside the cell. In general, receptors are Y-shaped or conical shaped. They funnel basically the material from outside the cell into the cell. We're going to see this Y-shaped general structure again with respect to protein, but in the shape of the antibody. So we'll be coming back to that. We have messengers, receptors, and transporters all here, and they're all involved in cellular activity. So whether that's a transporter that you see here in the glucose, that's a protein. Whether it's an insulin receptor, for instance, that's a protein, or whether it's actually insulin itself, the hormone, which is also a protein. Muscle contractions, right? So here is a cartoon of some of the different components of the sarcomere, or one of the segments within the muscle fiber. So the M line is in the middle, M line for M for middle. I guess you can remember that. And you have what's considered to be the Z disc, and that would be the boundaries then that would contain this actin, nebulin, myosin, and titin. Okay, so all these individual components. I give a description over here, and I say that the molecules of titin, which look like the spring coil, well, you know what? That helps keeps this myosin filament in line and centered within that sarcomere system. So this spring system, well, which I'm referring to as titin, almost acts as if it can steady the arrangement and the displacement of myosin as it slides back and forth, moving left to right horizontally in this orientation here. And it helps give it some boundary. The nebulin actually gives shape and determines the distance of that sarcomere length, whereas the actin and the myosin are what are going to be sliding against each other. So all of this is happens in muscle contraction and relaxation. Then you see in this next cartoon how you have actin further away from the myosin, but then whenever there is contraction, they almost perfectly are anti-parallel with one another in terms of actual distance. Again, what that distance is going to be based on the titin, the degree from which those fibers, those muscle fibers, which are made of protein, are going to be dictating what that strength will be. And of course, the nebulin is thought to act as, well, what is the overall size of that? So just kind of an interesting case there with respect to muscle contractions. You see that the titin and the nebulin also have additional roles, almost as if they're structure like proteins as well. So kind of interesting too, interesting as well. So you have this this globular region here of the head, almost looks Y-shaped in the way that it's shaped here. And this is the myosin 2 molecule, and you can see how it kind of like Y's out or V's out, I guess you can say, at the ends of these myosin filaments. They are comprised of alpha helical, segments all wrapped up tight. So it resembles that keratin and the fibrous kind of imagery that we had shown previously in webinar 2, where we had all these tightly coiled bundles. You can imagine that it's going to be fairly rigorous inside, that you're going to have strong intermolecular forces that are going to be able to keep the integrity of those helices. You will have minimal forces then wrapped to keep them together, and they will be able to repel water sufficiently so that they don't become disentangled and at risk for dissolution or just becoming denatured just in general. So it's pretty interesting. Of course, if the muscle fiber does get damaged, obviously, then there's going to be some problems, and then all of this architecture here will suffer accordingly. But this is just a great example of a muscle contraction and some of the structure where proteins are replete throughout this sarcomere system. So pretty interesting stuff there. With respect to storage, this is also pretty interesting. So here's a ferritin protein. It consists of about 24 subunits. So we had talked previously about hemoglobin, for instance, has the 4 subunits. Here in this molecule, this ferritin, you have 24 of these subunits. And so this is really interesting. Each one of these single units has about that kind of size. You see about 12 nanometers in overall length from the entire 24 subunits. So you can see how one segment of those subunits is going to be much smaller than that, maybe just a couple of nanometers. And you have the cartoon, which has the space filling as shown here. Like, again, there's more of a globule shape, indicating that there's probably some electron density right around all that stuff. If you don't really want to see all that, then the image on the far right just shows you where all those alpha helices are, and there's a ton of them, right? And in the middle is where you would have the iron. So some of this stuff is good for storage of iron-rich systems. Maybe that could be useful in building new tissue. In another case, you have casein, and we've already talked about it with respect to that one milk denaturation experiment with acids. And so I advise you to go back and review that again. But in the bottom left here, you have an SEM image of a globule of casein, and that's just what it looks like. And remember, this lacks tertiary structure. You can see how even in the ferritin, it all of the structures are just piecemeal, like kind of like a puzzle into itself. You know, is it really good for having tertiary and maybe even quaternary structure? Well, it all depends on what the purpose is for. If it's to deliver the components that are going to be useful in building new tissue, for instance, a mother to child transfer of milk, for instance, with the casein, you're going to want that, or one is going to want that, life is going to want that, evolutionary biological, you know, approaches are going to want that, because you don't want to have a protein that is too complex that is going to require a lot more action to be able to get to those vital nutrients. So in this particular case, the casein, for instance, would be able to deliver the calcium and phosphate. Remember, it's a 10-to-1 ratio, calcium phosphate to casein, so that the infant, the baby, can start to, you know, strengthen and further grow in response to what the nutritional needs naturally are. So in this example, you have the whey protein also intermixed with the casein protein. The whey in the bottom image here, where you have all these orange spheres, which really indicate that that's the casein. You have these solid spheres, the gray within that represent calcium phosphate within that, those casein globules. And then on the very end here, you can have some of these whey molecules, which are also proteins, right? And that's more the clarified stuff. That's more the lean and means kind of protein that you would want if you wanted to, for instance, build muscle, or just eat in a more lean way without the additional casein, because you know, if you don't need that extra storage type of protein for bone development or just general nutrient needs, then obviously you would reduce that if you'd like. Plus, you also know that that's more where the fat is going to retain as well. It's going to be very attracted to that. In fact, that is the basis for ice cream mixture, right? So you have these different components, right? You have the casein micelles. Again, these are all micelles. You have whole milk, which would generally lead to a better ice cream, right? So it's generally going to be fattier. It's going to be able to form those ice matrix, right? This 3D network as shown here in this right image. You even have fat interspersed in that. And all of this goes into trying to create, I guess, a good tasting, you know, texture rich kind of ice cream that would be highly desirable. If you're talking about like a fat free or a low fat ice cream, then obviously some different things are going to have to be done in order to try to get the texture profile and the melting profile, and I mean all that stuff in the right way. But as you already know, the casein can be readily susceptible to denaturation in the presence of acids, for instance, and in that particular case, and then the whey can separate from the casein. Okay, so there was that example. Here's another one, structure, right? Structure, function, dentin and skin. I'm going to show you here some images. This is some of my work that I had done previously. I had published this back in 2012, but this would be the top view look, and we're kind of zeroing in here on dentin that lacks all of the enamel. So the enamel has already been shaved off, just looking at dentin. And dentin actually has a little bit of calcium phosphate in it, of course. If it's considered sound or healthy, then you're going to have a bit of that calcium phosphate and other minerals and other agents like magnesium, for instance, some sodium. All of that will be collected around the opening of these tubules, because these tubules actually extend down into the, you know, further into the tooth, and eventually they get to the root, and there's going to be nerves. And if you demineralize or you weaken all of that mineral that had occupied those tubule openings on the right-hand side in images C and D, you get a close-up in D, and C, you know, it's further out. So so image D is like the tree, and then C is the forest. You see how the opening of the dentin tubule is, again, looking straight down on this. So pretty interesting structure. And I tell you what, you know, there's a really neat, one of my collaborators in Japan, he had put together this really interesting demo of dentin going straight down into the system. And it's pretty neat. You actually see the traveling within, and you can get an idea of how deep the dentin truly is. So if we just look at this structure, you'll be able to tell that, you know, what we're doing, we're looking at the side view, we're trying to dissolve that layer by layer. There's a neat technique. Here, we're actually probing down inside that dentin. Look how far it extends. So this is on the orders of microns. Look how deep these tubules are within a dentin substrate. So this is existing in your mouth, obviously, you know, a cow's mouth. I mean, anything that has a tooth that has the underlying dentin that supports the outer enamel layer, the biting surface, and that's what it does. So kind of cool, I think. And so you have there with the dentin that is collagen rich. It's formed from proteins. Here you have skin, and then you have the multiple layers. You have the epidermis layer, right? You have the dermis, and then you have the adipose tissue, which is underneath. So you have all of these different layers. Obviously, in the adipose tissue, you're going to get a higher content of fat, but the dermis and the epidermis is definitely made up of proteins. And you could even take a closer look at all this stuff. I mean, here's some type 1 collagen that's responsible for some of the structure. You can see what happens here in the assembly of this, and if it's directed, then you're going to get these nice uniform fibrils. If it's non-uniform, you're actually going to get some of this disoriented stuff. And so this can actually be synthesized, and this is part of the biochemical approaches, biomedical approaches, in trying to construct new bioengineered materials that can end up replacing, for instance, lost skin due to burning, or you know, maybe there just isn't enough skin to harvest on your body to replace that, grafting, you know, as it's also known, or maybe it's just one of these healing materials, like say you have a patient that is up in age, you know, your skin becomes less elastic as you age, just naturally. It just starts to lose water. So any kind of healing effect is going to take a lot longer, and it may not even be very good in terms of the end result. A lot of scarring might result, some stretching, you know, just some different things that are going to be unwanted. And so this would be some of the motivation to try to create a new bioengineered material that has skin-like properties, and it's of course then based on, well, how does type 1 collagen even form any kind of fibril that would lend itself to being some kind of skin-like material. So there's definitely research there. You can look at those links if you want, in case you're interested. But there's a lot of work going on with proteins. It's amazing whenever we see all of the uses of proteins here and realizing that we need essential amino acids, but yet we can't get them usually from our body, that we have to get those from the diet. This takes us back to the first webinar. How impressive is it then, again, with some of these bacteria that can just do this automatically, not only just make, you know, the essential ones that it needs, but even others that it can then use as metabolites in the course of making other valuable chemicals, you know, for whatever reason, including nefarious ones, you know, degrading the host tissue. So here's an example of antibodies. For instance, immunoglobulin, what that does is that helps destroy antigens. Basically, an antigen is a foreign substance that acts on the body. So this would be an example. Even if you were to undergo, I don't know, I mean, any you know, if you were to be exposed to any kind of virus or bacteria, or even certain types of, you know, cancers, right? I mean, a cancer would be considered to be a foreign substance. I mean, it's not natural. It has decided to, you know, that's whenever you have healthy cells that go awry, basically. And so your antibody counts will probably be pretty high. Antibodies can then typically try to destroy that, but they also, in the process of doing that, can also destroy your healthy tissue. So it's kind of a catch-22, right? You have to, you want to kill the insult in the body, but in doing so, there's going to be some collateral damage, perhaps. And so there's a lot of research right now on trying to figure out, and it has been going on for quite some time, how can we improve the disease-only trapping, killing, elimination, while maintaining, you know, hopefully minimally, you know, damage, a minimally invasive approach with healthy tissue. So it continues on. There's a lot of things that need to be done in order to try to do that. I mean, obviously you guys are seeing that the body is very complicated. We're just looking at all the different proteins, and these proteins are very, you know, amenable to being, I don't know, denatured, for instance, or modified, even even for good, depending on what that local situation is. Is it properly hydrated? Are there acids and bases? Are there metal ions involved? Is there localized heating? You know, I mean, all of this stuff can influence the behavior of these proteins, including, you know, for instance, pH. So here you have a Y-shaped kind of antibody, and here's a bit of a description. So this, you know, you're going to have a lot more. Notice how you have a lot more of these beta sheets, not the alpha helices, but beta sheets. I think that's pretty interesting. So that's, again, that's another type of secondary structure. Although we're used to seeing the alpha helix, here you have beta sheets where you have planes, like the folding of multiple helical patterns that have just been have just been not overlapped, but kind of wrapped around in a bendy way all along, for instance, like an XY plane. And here you have the antigen binding sites. In this particular case, you're going to see that the protonated amines are going to tend to the outside, while you have the carboxylates in the middle. And guess what? Everything is held together by disulfide bonds, right? So you have those linkages. And so you can see how this antibody is especially reactive, because look at all the charged environments that it has. You know, all of the ends have the carboxylate or protonated amine ends, the C and N, or N, I should say, terminuses. So if it's comprised of green and yellow, then you're going to have some polypeptides that are going to be not as many amino acids, but then the heavier ones of the polypeptides will be indicated by the red and blue. So you can see how the green and yellow, if they're the lighter ones, they're going to almost allow for a little bit more flexibility, right, and more spatial interaction, amenability to be able to help the bigger polypeptides respond to the agent that the antibody is gunning for, really. So pretty neat. This is the quaternary structure, and again, the quaternary structure is maintained by the disulfides, which we had already introduced there, and we do still have some coils here as well. And remember, the coils do have that helical shape, and so there can still be maybe a bit of hydrophilic, character with that, hydrogen bonding, a little bit of that polar character, but definitely, yeah, the hydrophobic or the non-polar aspects are going to be realized as well. Okay, finally, we're going to talk about catalysts. All right, and this is a pretty important aspect of proteins, right? And we need enzymes to help make a reaction go more quickly than it would otherwise. So as this definition here, and we're going to consider them to be the proteins, for instance, hexokinase, they accelerate metabolism and remain intact, all right? So for instance, the first step in glycolysis, if you have some ATP based structures, right? You have a little bit of glucose, and what's going to happen? How's glucose going to turn into glucose 6-phosphate? How is that ATP molecule here going to be divided up? Well, you're going to need some hexokinase to be present. So the hexokinase helps to detach one of the phosphate groups, right? Attaches it to the glucose, and then you're left with ADP. So which one is larger? Well, ATP is larger than ADP, right? So you have a reduction in size here. The hexokinase help make that happen. So in this particular example, you have substrates being the reactants, and that's always going to be the case. But in this example, you have hexokinase and glucose responding to one another. So in this case, the glucose is a reactant, as is the ATP. But here's the interesting part, right? Reactants can be specific to enzymes. For instance, the D-glucose, not the L-glucose or D-galactose, are going to be able to respond with hexokinase. Okay, so this is known as substrate specificity. So if certain substrates are going to be reacting with certain enzymes, then, and I call this like, with respect to an active site, which is going to be like a bespoke opening. It's almost like a glove that's going to fit perfectly in that hand. It's going to develop in that enzyme to be able to accept that desired substrate, whether that substrate is D-glucose in this particular example or something completely different. then, that will depend. But in this example, you have the hexokinase, which is this green enzyme here, and within that opening, you have a bit of glucose being admitted into that active site. And this is the close-up of that site, and it shows you that there are quite a significant amount of hydrogen bonding going on in order to help maintain that substrate enzyme connectivity. Cofactors are sometimes present. These are non-protein helpers. Usually they're inorganic in nature. Otherwise, we talk about agents that can help out as being coenzymes, and these are also helpers, but this time they're organic, like FAD, okay, NADH, things like this. Those are all coenzymes, and they work together with that enzyme in order to make the reaction occur. Note the size of the enzyme. Look how large it is compared to the substrate, okay? It's massive, right? So this begs two different models then, or lines of thinking. They've been separated by about almost 50 years apart from whenever they were initially introduced. I think in the late 1900s, this lock and key model was introduced, and then there was another one that was introduced called the induced fit. That kind of is a better fit for what most of the enzyme action is going on in our bodies, at least. So the lock and key just says, look, you have a substrate of a particular size and an enzyme of a fixed, receiver size, and they're going to come together, and that's it. That's nice for specificity, but it lacks in its ability to adjust for transition states that may occur whenever the substrate and enzyme come together. For instance, this is nice. I mean, this puzzle piece kind of model is really nice, but what happens if there's an intermediary transition state, which there often is? And so that's why the induced fit model was introduced. And that says, you know what, the substrate can be what it is. If you look at the green substrate piece here, both in A and B models are both the same, but look at the enzyme, it's a little bit different, right? So the enzyme flexibility is greater in the induced fit model. It says, look, the enzyme doesn't have to be perfect because it's really hard to get all these perfect enzymes to exist, knowing that at any given temperature, you're gonna have fluctuations in that. It can, solvent, amounts, pH concentration. In other words, what is the local environment? What is the, what is the concentration of ions present or nearby? All that stuff matters. And so the induced fit model kind of says, well, look, we know that the enzyme and the substrate can come together, and they're going to be able to, lock in and make that reaction happen, but the enzyme will have a bit more flexibility. So in this case, if you were to ask, well, looking at hexokinase and D-glucose, which model would be a better fit? It would be the induced fit, because the enzyme, again, this hexokinase, look at the size of that molecule. Is it going to be perfectly rigid to be able to accept that glucose substrate as is all the time? No, it's going to be shifted. It's going to be dependent upon local environments, changes in local environments. And when I say that, again, it's about concentration of any kind of solvent amounts, pH changes, temperature changes, just things like that. So here you have this kinase molecule. Before it admits that glucose, upon admitting it, it kind of wraps around it a little bit. Again, that's some of that in between, transitionary feel and flexibility that the induced fit model allows for. What are some factors then that influence the rate of enzymatic reaction? Well, we have a couple of them. The very first thing, and this is a rule anyway of chemistry that we had talked about even at the beginning of the semester, we said, look, they need to be able to come together. Your reactants need to be able to interface. So the proximity of these needs to happen. That's why solids are what they are, whereas liquids are what they are, and then gases. Gases are treated to be having a poor proximity of one another. And so unless you have, contained vessels that are very closed and you have other parameters that are being forced upon in order for that reaction to go, for instance, you generally are going to have heat because that's going to accelerate the gas molecules in order to get them to react. Gas phase is typically easier to react than, for instance, two solid phases. And you can imagine, you take two stones and you put them together, they're not really going to react too well. But if that stone was to become a gas somehow, like if you were going to sublime it at the higher temperature, you can imagine that those ions or atoms, would then coalesce into something new. So proximity is definitely important, and the orientation too. So I think that that's important as well. So this is what we mean by that. So you have substrates, orange and green. Okay, we're going to consider these both to be substrates. Okay, so this isn't just a one enzyme and one substrate model. These are going to be two substrates, and E is going to be the enzyme. You're going to have the orientation and the spatial separation of them. The proximity and orientation matter in how they're going to react with one another. And an enzyme helps to lock that together. So I think that that's pretty self-explanatory, all right? And then the other part is going to be the bond energy. What's really interesting here, and this is where these cofactors or coenzymes show up and they make a difference. So whether you have like magnesium ions or whether you have FAD or NADH, whenever they start appearing in an enzymatic process, they generally do so in order to allow for the reaction to be energetically favorable. So for instance, in this example, magnesium is locked on to two of the phosphate groups here. Everything is within this enzyme, right? So it's all part of the substrate. And we're talking about, for instance, glucose, right? That's the orange, hexagon here that comes close to this ATP model, right? Or molecule that has the phosphate groups. Magnesium, if it's present, it helps hold those phosphate groups together just long enough to be able to encourage that oxidation of the glucose, right? And so it's just an example of that being able to be possible by virtue of these cofactors. And again, also the coenzymes can do the same thing. Another factor then, so you have proximity, you have orientation, the presence of, for instance, cofactors or enzymes, which can affect bond energy. You also have how much of the substrate concentration. So for instance, here's the reaction rate on the y-axis, and then the substrate concentration goes on the x-axis. As you move from left to right, the enzyme activity will increase, but it will saturate at some point because there's a limiting factor anyway to how much reaction can proceed, given that there's going to be an upper limit of a substrate. So at some point, you can't get any more product that's going to be formed. And that's a case then where you reach what's considered to be a steady state. It's a condition of maximum activity. All of the enzyme is combined with the substrate. So the enzyme's pretty much tapped out, okay? And everything, you're not going to get the reaction to occur any more quickly than what it had been. And in fact, as you get close to the steady state, you can anticipate that the enzymatic reaction, right, is going to slow down. That's just going to be natural, okay? So what are some factors influencing enzyme activity? Another one, we talked about already 4 of them. How about pH? This is another one. For instance, you will find that there's going to be some enzymes like pepsin that can exist in the stomach, and they attack the peptide bonds. They help us break up those proteins in the stomach. Remember, in the stomach then is where digestion of proteins will occur. Carbohydrate, that's going to begin in the saliva. We'll talk about that in chapter 12, but just wanted to, this is a really nice indication, like point here, where if we're talking about pepsin in the stomach and the peptide bonds, where does digestion occur? The pH here is going to be 2. And so pepsin is really going to be an enzyme that loves an acidic environment. And it is secreted in the stomach, okay? And it helps to break up some of those proteins. You have sucrase, which busts up sucrose, small intestine, okay? So for instance, if you don't have a large intestine, then you won't probably be able to break up certain polysaccharides that are going to be able to be taken up, reabsorbed from the colon and into the bloodstream and whatnot. Sucrase, for instance, it is, something that is produced in the small intestine. It can bust up sucrose. The pH there is about 6.2, as you know, because we already introduced some of the digestive tract previously in some of the other webinars. So this is really interesting. So for instance, like my wife, she doesn't have a colon. She has to have limited amounts then of any kind of polysaccharide, but she has no problem eating, sugar-rich foodstuffs, including, cereals, just simple cakes, things like this, because her small intestine is fine. And it has no problem secreting, for instance, the sucrase enzyme that will help bust up the sucrose molecule. So just as a pretty interesting, and practical application of this. Here you have hexokinase. All tissues do this primarily, to be able to take in glucose and transform that into some kind of energy, right? So the pH there is 7.5. Urese is, secreted in the liver and it helps bust up urea. pH there, 7.4. Trypsin, all these kinds of things. Look, small intestine's also helpful for proteins, right? So the arginase in the liver, helps to bust up arginine. pH there is 9.7. Okay, but again, it's pretty interesting. Even in the small intestine, you might have lactase, and that's where that would occur. If you have lactase, and then you'd be able to bust up the lactose molecule. If you're lactose intolerant, then your small intestine probably lacks the lactase. Okay, that's funny. It's a lot of lacking, I guess. The amylase that we had talked about with the BINO, where might that exist? That might exist in your colon. Okay, so that is so important for breaking down some of those polysaccharides. So in the large intestine, you also have some enzymes that can help bust up fatty acids. All right, some fat, some fried foods. You're not going to be able to get a lot of that bustedness, or that, I'm calling it that. Basically, it's hydrolysis or decomposition, I guess you could even say. Within the small intestine or the stomach. That stuff is going to occur within the colon. And you already know that there's a ton of bacteria that resides in the colon relative to the stomach, for instance. So you can easily see how some of these enzymes are not just created by our own bodies, chemically, but it's more of a biochemical creation. It's an offshoot from the effect that we have because we have, just billions of microorganisms that live within our system. So, and that's obviously gonna help improve our digestion. Okay, so for a really cool demo, and I really want you guys to watch this one. This one's really cool. If you like the other, in-class demo that I did with hydrogen peroxide, I think you'll really enjoy this sweet potato one. I did a sweet potato versus hydrogen peroxide. Sweet potato is it's among all those plants. Okay, so there's lots of plants and there's, animals and plants and even bacteria. We, I should say, we, because, this is us included. We're a type of animal, obviously, but we manifest enzymes that can break down and attack hydrogen peroxide. And a sweet potato is no different, okay? It's just, I like to use the sweet potato because it's rich and it has great color. And when I say it's rich, it has ample enough of catalase, and I like the color, and it's easy to get large amounts of it in just having one vegetable, right? Anyway, catalase is pretty similar to hemoglobin. It's pretty interesting. Both contain 4 protein heme groups with the iron, as you remember. So it's probably no surprise then if iron was responsible in hemoglobin for attaching to oxygen, that guess what? Catalase then can help scavenge hydrogen peroxide by virtue of the iron's ability to try to lock on with the oxygen component of hydrogen peroxide. So these are just some still shots that I have of the video, but again, I want you to watch that. Start with the sweet potato. We just cut it up, and then we processed it in this crusher, slicer kind of thing. We then stuffed it in these centrifuge tubes. I had 6 of them, so we just filled them up. I think they're about 15 to 20 mils. Anyway, filled up the tubes, and then we centrifuged them for 10 minutes at 4,000 RPMs. And that was the endpoint result. So you get a liquid in the very top. So here it's almost about 4 mils of liquid. Notice how it's a little bit denser than the pellet, what's called the pellet, which is around 8.5 mils on down. That's also just the solids that weren't in so-called solution, which is the supernatant here with that lighter colored stuff. And in that lighter colored stuff, or I should say in that darker orange colored solution, the liquid solution, that's where the catalase is going to be. And we can take that and we could then put that into a glass tube. And then I did that for all of the 6 vials. And then I also added some hydrogen peroxide. Go check it out, and hopefully you'll enjoy that reaction. I don't want to spoil it, but pretty neat reaction. What's another factor affecting enzyme? Now we're getting into the negative effects. Okay, so temperature. if you are too hot, you don't work nearly as well. And that's where we say here, and I'm using these images from your book, the hot, sweaty workers. Sounds like a bad story, right? But you have hot, sweaty workers. Think they're going to be happy? Do you think they're going to want to work? They have all these parts. Are they really going to use all these tools and keep a happy profile and be well rested and create all of these bikes? No, they're not going to. Not if you don't give them air conditioning and plenty of fluid and everything else. So you know what? Enzymes are no different. So I did that experiment with the sweet potato, and I just used room temperature stuff. If I were to boil those potatoes first instead of using just raw, I would get a different outcome. Okay, it wouldn't be as pronounced because enzymes are not going to want to have excessive heat. The proteins, right? They unfold. You already know from some of the eggs and the milks that even if they lack tertiary structure or even quaternary structure, they're going to start to denature with respect to elevated temperatures at the very least. So it's no different here. So temperature can be an enemy of enzymes. That's one of the reasons why, if anybody's doing any kind of biochemical assessment of a body, right, it's important to get stuff done quickly. Like if I were to use that sweet potato, I have to hurry up and use and prepare that material in less than 30 minutes. Otherwise, the catalase activity is going to oxidize it's to the open air, and the effect that it's going to have on peroxide is going to be significantly reduced. If I'm trying to think of another instance, but that's just the way it is. It's like having, you know, if you open up a brand new bag of chips, right? Even Pringles, and if you don't eat them all within a certain amount of time, they're gonna go stale, right? So the oxidation's there. So it's no different. Okay, so you got to keep the enzymes in a healthy state and in a nice state that they can actually continue to do their work. So if medical samples were collected, they generally are going to put those on ice or in a refrigerator, keep everything cool until it absolutely needs to be tested, and then it can be tested. So if people are being tested for a virus or a bacteria or anything else, sometimes you're going to have false negatives, because if the samples were collected and then they were allowed to sit at room temperature, even for an hour, sometimes that's too long and under, you know, room temperature conditions. So if those samples aren't put away in a cool environment, you know, then some of the end diagnosis based on the laboratory results are going to be fudged, basically, because the conditions in which the extraction and preservation of that biological material wasn't optimum enough. So I guess that's pretty clear. So we also have inhibitors that can show up. All right, so these are other molecules that might look like a substrate that the enzyme wants to come around, but it isn't. And so these can either be considered competitive or non-competitive. So reversible inhibitors, competitive or non-competitive, and what they do is they try to compete then with enzyme, you know, with that seat at the enzyme table, as it were. Okay, so you have the seat that is one seat available. For instance, we're going to call E the enzyme, and that's the table. Kind of looks like a kind of an upside down, like kind of Pac-Man kind of thing. Anyway, that's the table, and there's one seat available, right? And the substrate is supposed to be sitting at that table, but then there's something else. We're going to call that the I. It's going to interfere with it. It's an inhibitor. It's going to pull up its seat first by virtue of maybe it was in higher concentration. Maybe it just had a greater proximity. Whatever be the case, it can do that. Here's an example. Like your liver produces an alcohol and dehydrogenase, an ADH. It also does an aldehyde basic dehydrogenase enzyme, which can also oxidize ethanol. But it has this this enzyme, and it responds to ethanol. But guess what? Ethylene glycol or methanol can also pull a seat up at that enzymatic table, and these would be considered inhibitors. I put them in blue for inhibitors. Orange is what you want, right? Alcohol, again, you guys probably remember, it oxidizes to a carboxylic acid. The interim step could be the aldehyde. ADH takes it to an aldehyde, and then there's another aldehyde-based dehydrogenase that takes it from the aldehyde to the carboxylic acid. Okay, so this is actually a two-step process to get the alcohol to a carboxylic acid. But it's no different than opening up the wine, right? So the wine, which has alcohol in it, that can oxidize to the acid. You know, first aldehyde, potentially, you know, potentially the aldehyde, but definitely acidic, and then it just doesn't taste as well. This is one of the reasons why methylene glycol, methanol, you know, if that does get put into the body, there's enzymes that receive that. How are you going to counter that? Well, you have to have a slow IV infusion of ethanol for several hours. So actually got to give ethanol because you want to increase the substrate concentration so that it can overwhelm the inhibitor's concentration. So then the enzyme will be attracted to that. So you can imagine how this is going to be a slow process. And you know, you can't obviously, you know, pass everything through the digestive system in this case, so you have to use an IV. Okay, so, you know, plan on being there for quite a while. So if somebody has ethylene glycol poisoning or methanol poisoning and they're admitted into the ER, well, you're going to have to hook up a drip, and the drip is going to be basically an ethanol drip. So yeah, it's one way, I guess, of getting your alcohol fix. All right, but what happens if they're non-competitive? Hey, it happens. So you have the seat at the table, right? And you know, you have the substrate that wants to be there. But what happens if another seat all of a sudden is just, well, it's carved up, somebody else completely different, right? It's a non-competitive, completely different, unique kind of substrate wants to come in. You don't want it. It's still an inhibitor, but it makes its own space at the table. So it's the neighbor that nobody wants, right? That says, hey, I'm going to make my home here, right? It's almost like a chipmunk, right? If you have too many chipmunks around your home, right? I mean, they're just going to do their own thing. They could sit at the table, right? They could just use exactly what they want, but they don't. They want to go up into your attic or underneath your foundation or whatever and make their own home. They're carving out their own seat, okay? So that would be the example. In this particular case, you have this echinocandin. I think it's pretty neat. This is a class of lipopeptides. These are water soluble. This would be a good example of an inhibitor, right? And what it does is it distorts the 1,3-beta-glucan synthase of Aspergillus species. So it disrupts cell wall action, okay? It is actually considered the penicillin of antifungal. So a fungal species rely on beta-glucan synthase, okay, that's an enzyme, to create their beta-glucans, which is their membrane. It's basically a cellular membrane. Humans don't have that, by the way. Then this echinocandin can interrupt that. Then you can have a method of controlling fungals. So here's a good example of a class of antifungals known as echinocandins, and what it does is it creates its own table. It's like that, you know, chipmunk coming in and it's saying, look, I know that there is a competitive inhibitor that could sit at that table, but you know what? We're going to create our own, and it's still going to work, and it's going to work the way that we want. So it's a pretty interesting way of approaching a problem using, I don't know, I don't want to say an unconventional approach. We will say non-competitive approach, but there's a lot of action there trying to understand, well, how can one species interrupt another species, and can we make use of that? So if there are antifungals, antimicrobials, right, antivirals that are being made, sometimes these are based on non-competitive inhibition of enzymatic activity, and this would be an example of that. Another example of this would be the inactivation of an enzyme, okay? So for instance, here you have an inhibitor example where we're going to use a transition metal like silver, mercury, or lead that bonds to the diol of amino acids. That interrupts them, so that's toxic, right? You don't want that. And or copper, for instance, you saw how the egg white turned to slime. That would be an example of an inhibitor, and it would be irreversible. You can't get the copper back. You can't really extract out that silver or mercury or lead, for instance, if it was bonded to that diol or even that protein. Once it's done, it's done. You just gotta let that run its course. You can use an emetic if you need to vomit that out or whatever, if it was a systemic thing within the body. But that's just, you just kind of have to, or, you know, just natural, you know, processing the other way, I guess. Obviously that could be, you know, an uncomfortable, unhealthy situation, potentially even dangerous situation. And then here you have another example, not only with the transition metals that can lead to that denaturation of proteins via, for instance, the formation of sulfides because it interrupts the, you know, those sulfide linkages. but you also have something that could be good. So here's where penicillin binds to bacterial walls. Again, the beta-glucan. So just like how you had that echinocandin class of antifungals that did essentially the same thing, penicillin, which is produced from mold spores, right? From moldy fruit, can actually attack to certain bacterial walls. And this is one of the first lines of antimicrobials, antibacterials that was developed, right? And because it's not found in humans, then we don't have to worry about it affecting the human bacterial walls because we don't have the beta-glucans, but the bacteria does. So if you're looking to kill the bacteria, well, this is the first line antibiotic that has been used to be able to do so. So pretty neat. And this is the structure of the penicillin molecule. You can even see the carboxylic acid, the carboxylate group right here, right? The N group right there. And you have carbonyl here, right? So that could be part of the ester that's going on. And R is a variable group, but it's that action of the carboxylate that is able to interact with the beta-glucans of the bacteria. And so that is it, everybody. So that wraps up chapter 10. So I hope you're able to get your work done, but reach out with any questions and we'll go on from there. All right. Thank you for listening and watching.